Welcome to the Gary Price Memorial Lecture 2014. If you can cast your mind back to 1982, which may be a bit before some of you, I met Gary Price in Norman Bowery's lab. Gary came to do a PhD in 1982, and we were working on a novel GABA receptor that came to be known as the GABA-B receptor. And we became friends, and Gary went on to do a postdoc at Roche, and then another postdoc at the Hammersmith cyclotron unit. And meanwhile, I went off to Bristol, the psychopharmacology unit, and in the late 90s, Gary was working for what was SmithKline Beecham, and then he climbed the ranks to a director at GSK, and he sponsored some research that I was doing, and we became close again. That research also involved him coming to do some undergraduate teaching. So at the time I was working for David Nutt, I'm sure you all know who David Nutt is, and I pushed for Gary to become visiting professor of psychopharmacology. So I did the paperwork and it was signed off, and Gary was very proud to be a visiting professor of psychopharmacology from about 2001, I believe. We're very grateful to GSK who actually sponsor these talks. They put up a fund to support the speakers, so we've been going for several years now. Norman Bowery was the first chairman of the committee that organised these talks, and he actually gave the first talk himself in 2003. And then we had other eminent speakers like Hans Müller, David Nutt, Tom Blackburn, I don't think Tom's here today, Ian Martin. And then Norman Bowery then retired, and he asked me if I would take over chairmanship of the committee. So I was quite anxious to take over this committee because we'd had such good speakers, but I chair a small committee now that meets with previous speakers by email, and we get together to organise the next speaker. And so the first one I organised was Anne Stevenson in 2008. Then we had John Peters from Dundee, Annette Dolphin, Sarah Lummis, David Wiley last year. And this year, we've come up with a real treat for you. We have a well-known speaker who obtained her PhD back in 1988 from Barts Medical School. She then went on to do a postdoc at UCL, University College London, before getting a lectureship at the School of Pharmacy, that's the one, yes. And she is now at University College London as the A.J. Clarke Professorship. I'd like to present to you Professor Lucia Sivilotti. She's had many great publications. She's published with David Calhoun, and I think they had a Nature paper some 4 or 5 years ago. She's very well published. And Lucia is going to talk about agonist efficacy, the view from the single receptor. Thank you, Lucia.
Thank you. Thank you, Alan. It's a great honor to be here, and it's a privilege to be able to review the work of my lab and to be able to tell you about our work. What we do, we record the single channel currents, the currents that come from the activation of single channels in ligand-gated ion channels in the nicotinic superfamily. I will show you how doing this sort of work allows us to measure the molecular steps in the activation of the receptor by the agonist, and how we can measure in this system agonist efficacy and pinpoint which of these steps in receptor activation are the determinants of agonist efficacy. Now, I'm a pharmacologist, speaking to pharmacologists, and the next slide is completely superfluous. You don't need me to tell you that the most approachable of physical constants that we can measure as pharmacologists does away with agonists. It's a binding affinity for a competitive antagonist. So it's competitive antagonists that are the pharmacologist's best friend. A.J. Clarke in 1926 and John Gadam showed that a competitive antagonist produces a shift in the dose-response curve of the agonist. And if we measure this shift at dose ratios, Heinz Schild, in my own department at UCL, showed that from the analysis of these dose ratios, from the Schild plot, and after all, the Schild plot is even in the address of our society, isn't it? You can get the affinity constant for the competitive antagonists. And the thing that is interesting here is that we've done away entirely with agonist complications here. We've eliminated it. That's the beauty, the power, and the elegance of the null method. It's done away with all the cascade of complications that are linked with agonist efficacy. So agonist efficacy, we entered there at our peril, of course. So agonists are difficult. This is a milestone in pharmacology. This is Stevenson, a figure from Stevenson's paper, 1956, showing the dose-response curves to a bunch of agonists. And in order to characterize an agonist, we need to measure its potency, its position of the dose-response curve on the horizontal axis. It's EC50. But we also need to measure its maximum response, and that can be different for different agonists. So we need to measure the potency and the efficacy of the agonist. So in this dualism, we can just look at it and say, well, potency and efficacy, are they distinct concepts? Are they different concepts? Are they different properties of the agonist? Well, a year after this, in 1957, José del Castillo and Bernard Katz, in the biophysics department at University College, published another paper on agonist efficacy. And it is this paper that has more or less set, in 1957, before I was born, the agenda for the work on agonist efficacy of my sort of agonist efficacy in the ligand-gated field. This was a paper, the Del Castillo-Katz paper in 1957. Why is this paper such a milestone for us? Because they chose to work on ligand-gated ion channels. In ligand-gated ion channels, frog neuromuscular junction in that particular case, you can measure very directly the agonist effect, the activation of the receptor that is a consequence of agonist effect. We were reminded brilliantly yesterday by the Gadam lecture how complicated it is, the concept of efficacy in GPCRs. Here we are in ligand-gated ion channels. And the power of shifting to ligand-gated ion channels is that it allows us very much more direct measurements of the physical reality of efficacy. The other key point that Del Castillo and Katz had was that they were willing to speculate and hypothesize a precise physical mechanism for the action of an agonist. So these are the words that they wrote. The receptor, we didn't know what the receptor was then. Probably didn't even know it was a protein. It reacts by a two-step process, forming an intermediate inactive compound which is then changed into an active depolarizing form. And they wrote down this Del Castillo-Katz equation. Now, what does this mechanism? You see, you have an equilibrium for the binding and then an equilibrium for the gating. What does this mechanism predict in terms of dose response curves? Now, let's write it how I write it for my students at UCL. The agonist, A, binds to the receptor R. There is an equilibrium, a binding equilibrium, that is described by the dissociation constant for the agonist on the receptor. The bound complex then can open, and this is described by E, that takes its name from efficacy. So, can we get a dose-response curve out of this? What does this predict in terms of the dose-response curve? Now, first of all, the maximum response. If you measure the maximum response in the best possible way, it can be done. It can be tricky, but it can be done. I'll show you later. That is, as maximum P open, the maximum response depends only on the second step, on E. But what about EC50? What does EC50 depend on? EC50, it turns out, depends on both steps. It depends on both steps in the equilibrium. It depends on the initial binding, and it depends on the efficacy of the subsequent gating. So we have KD over E plus 1. Apropos of this, I can do no better than to send you to one of the most cited papers, most cited reviews in British Journal of Pharmacology by my mentor at University College London, David Colquhoun, who I'm glad to see here. This is binding and gating. This is a binding and gating problem. How can we distinguish these two steps? Now, David, in 1998 here, was in a very good position to comment on this because he was the first to be able to measure actual agonist efficacy in the frog neuromuscular receptor, acetylcholine-nicotinic receptor. Again, a strategy similar to that used by Bernard Katz. What did he do? He was collaborating at the time with Bert Sackman, who invented two inventors of patch clamp. He was in Göttingen at the time. You can see here, these are single channels, single molecule recording. So closed, and then the channel opens in response to acetylcholine, closes again, and you see bunches of openings by the different agonists. The analysis that Colquhoun and Sackman did, this unfortunately is in J-Physiol, it should really be in BJP, but there you are. The analysis that they did analyzed the fine structure of these groups of openings and measured for the first time E, or efficacy, and for acetylcholine this is very high, 43. 43 efficacy means that when the channel is bound to acetylcholine, in the best possible conditions of measurement, you should see that it is open 43/4 of the time, that is 98% of the time. So acetylcholine is an extremely efficacious agonist. And these results have stood the test of time. But note, I keep telling you about the maximum P open, but these guys here didn't measure the maximum P open. The reason they didn't do it straight away is that it's tricky. The muscle nicotinic receptor is a great receptor to work with. It's quite a robust one. It expresses very well. It's consistent. But it has got a little nasty property, and that is that it is a cationic receptor. Its channel is permeable to cations, and acetylcholine, which is a natural agonist, neurotransmitter, is a small cation. So acetylcholine not only activates the channel, but it can also block the muscle nicotinic receptor. And that is a technical complication. We can deal with it nowadays, in 2014, with the new analysis programs that we have and with technical improvements and with other tricks, as I'll show you later. But in those days, they worked it out at the level, at low concentration, with low concentration work, and they couldn't get a dose response curve in the first instance. So it's clear that it would be a nice thing if we have a different receptor to work with, a receptor that doesn't have these problems. Now, I will switch now from milestones in history to my own personal story. And the next chapter in the story sees me in David Colquhoun's lab at University College as a postdoc, and it is the story of a great piece of luck, a great stroke of luck. I found that another receptor, a receptor related to the nicotinic receptor, the glycine receptor, it's an inhibitory receptor that is very important for fast inhibition in the spinal cord. And I'm sure that some of you have been at Trevor Smart and Rob Harvey's symposium on Tuesday and heard a lot about this receptor. And it is an ideal receptor to do this sort of work with. How did I stumble into this receptor? Well, somebody at University College Hospital Pediatrics Department, Michelle Rees, diagnosed a child with a channelopathy. The child had Startle disease mutation. A Startle disease was already known to be due largely to mutations and loss of glycinergic transmission due to mutations in the glycine receptor. Startle disease does exactly what is written on the tin. That is, the patient, it's not a particularly serious inherited channelopathy, can be controlled reasonably easily with clonazepam, but the patients react very markedly to trivial sensory stimuli and habituate with difficulty. So it was already known in those days that it was due to mutations in the subunits of the glycine receptor. Now, I'm going to treat you to, this is a vintage 1997, one of my slides, vintage 1997, and you can see what we knew of the receptor subunits then. Receptors in this superfamily are pentamers made of 5 subunits of this sort, and this is a topology as we knew then: amino terminal, piece of string or blob, extracellular, and then 4 transmembrane domains and the amino terminal. This is a membrane. Now, we did not know where the binding site was. We kind of reckoned that it had to be in the extracellular domain because most neurotransmitters for these receptors are polar molecules, and they couldn't go through, certainly, the membrane. But at the time, these were the mutations that were known to cause Startle disease at the time. This is our own here, K276E. But at the time, it was thought, it had been hypothesized by the leading lab on glycine receptor, that of Heinrich Betz at the Max Planck Frankfurt, that because we were seeing a profound loss in potency of glycine, of these channels, of these mutated channels, the loss of potency was due to a loss of binding. And therefore, these mutations were in the binding site. Well, what did we see when we expressed the mutant in oocytes? So this is another of my old slides. And you can see these are the dose-response curve that you obtain in an oocyte for wild type, 200 micromolar EC50, and this is a mutant. We see that we have a profound shift to the right. This is 5.4 millimolar glycine EC50, and we also have a profound loss in the maximum response. Of course, the maximum response, this is not a p-open maximum response, so I don't know, it could very well be that maybe this mutant is also not expressing so well. So while this loss of maximum response speaks very much in favor of a gating mutation, of a loss in the ability of the channel to open in response to the agonist, we weren't initially quite sure. So what can you do to prove that out? Well, we could try to look, for instance, at the binding site. Now, how can we look at the binding site? Well, we're pharmacologists, so what we can do is do a Schild analysis and see whether the competitive antagonist strychnine still has in the mutant the same KD. If we see similar KD, this will argue in favor of the idea that there isn't a great change in the binding site for glycine, because glycine and strychnine must overlap in the binding site. And so this is, this is meat and potatoes to us. This is a Schild plot, and this is a KD, and you see the KD is not changed by the mutation at all, still about 20 nanomolar. What I also did was I obtained single-channel recordings from wild type and from mutant channels, and I could see 2 things. One, that the channel was less stable in the open state. The openings were much shorter. And the second thing, which at this point in time is the most interesting to us, this channel was an ideal channel to work on, a wonderful channel to work on, the best. So I'm going to show you now what happened after I set up my first lab at the School of Pharmacy, where I was appointed by Trevor Smart in 1998. And it took us a while, but we got quite a lot of excellent data on the glycine receptor activation. So what does the glycine receptor look like as a single channel? Now, more single channel recordings. This time, the openings are upwards. You can see this is a shut state here. This is cell attached, activated by a very low concentration of glycine, 10 micromolar. And the upward strokes here are openings. Some of them are so short that they look smaller, but they are all the same conductance, because this is a cell attached recording. And we can see that the openings at this low concentration occur either in single openings or in groups of few openings, what we call in jargon a burst. Now, what does this mean really? The channel is shut, then the channel binds the agonist, the transmitter glycine, and while glycine is bound, the channel can flicker between closed and open states. But when glycine dissociates, the channel has closed, glycine dissociates, and then until another molecule of glycine rebinds, the channel cannot open. So this shut time here between these two bursts is a reflection of binding process of glycine to the channel. So this is somehow linked to agonist affinity. You will see that that is true because if I increase the concentration of glycine, you will see that these intervals become shorter and the bursts start running into one another. You see here, the bursts have become much more frequent because these intervals are shorter. You see also another thing. There are long shut times here. These are the molecular, the microscopic equivalent of desensitized states. And you can see them, we can cut them off. We can eliminate them from our analysis, so our analysis is going to be confined to the activation of the receptor, to binding and gating in a very Del Castillo-Katz way. When we increase the concentration of glycine to the top of the dose-response curve, this is what we get. We get that the channel is either desensitized or it's open all the time. So you see the P-open of the glycine channel, opened by glycine, is very, very high. It's almost 100%. And this beautiful channel doesn't have agonist self-block. Now, I was so lucky. If I had tried with another of the isoforms of the glycine channel like alpha-2, goodness knows what I would be doing right now. So this was an enormous piece of luck. This is a synaptic alpha-1 beta glycine receptor. The best receptor, has all my gratitude. So, how do we get a dose-response curve out of this? We're pharmacologists, come on. Let's get a dose-response curve. Well, we record, again, at a lot of concentrations. We cut off the desensitized states, and we measure the P-open in here. Yeah? This is a P-open. This is a response at single-channel level. And this is the most beautiful of all dose-response curves, and certainly the most interpretable. This is a clean dose-response curve that is at a one-molecule level. So this is the expression of binding and gating by the agonist. There's no desensitization here, there's no spare receptors, no complications. So this is what happens with glycine, which is a full agonist. So this was 2004, and these are the data by my first PhD student there, Valeria Burzomato. But can we look at a partial agonist? What would a partial agonist look like at single molecule level? Well, we had to find the partial agonist. No, it's now 2004. The next step was published in 2008. We found a good partial agonist in taurine. So the agonist had to be just right, like Goldilocks, you know, not too full agonist, not too partial. The other thing about taurine is that taurine, unfortunately glycine is a great stuff, but glycine is a contaminant that is present in a lot of solutions, and we've got to be very, very careful to get rid of it. If you're working with taurine, you need concentrations as high as 100 millimolar, and these contain a significant amount of glycine, an amount of glycine that is such, concentration that is such, that will ruin your work. So it's only because we identified this by HPLC and because David Jane in Bristol very kindly purified this to better than 1 part in a million impurity of glycine, that we could do what I'll show you now. And what I'll show you now is what a partial agonist looks like at single molecule level. This is it. Taurine, 100 millimolar, is saturating the receptor. There is no binding site that is empty, but taurine cannot keep the channel open all the time. it opens it 50-ish% of the time, 54% of the time. So these two dose response curves are the full agonist and the partial agonist. So as you can see, 4 years have passed. So this is not a fast way of getting data. So this is taurine. How do we move from here? Well, what we need to do now is to try to understand what is happening here and analyze our data so that we can connect them. I was telling you, the molecular steps in activation. What we need to do, in other words, is to write down something that is an up-to-date version of the Del Castillo-Katz mechanism which answers all the questions that we want answered. And those are, we need, how many binding steps are there? How many gating steps are there? How are these steps connected? And most importantly, we must quantify the rate constants that connect these states and the equilibrium constant, or the ratio of rate constants. Now, what we did was made possible by advances in stochastic theory of single channels, made possible by the work by Alan Hawkes and David Cahoon in the 90s, and in particular to the process, to computational processes that allow us to fit models directly to the recordings. Now, what we do is also test a lot of mechanisms, and we validate the mechanism against the data. I won't have very much time to show you. I will show you just one validation, but we ended up in 2004 discarding about, I think it was 34 models tested and one survived. So, what does the model look like? It's a little bit more complicated than Del Castillo-Katz, but not that much. Now, we have a pentamer, remember, and we have probably three alpha subunits and probably 3 possible binding sites for the agonist. So we have another mechanism, so R binds, this time it binds 3 molecules of agonist, glycine or taurine, as it may be. So this is a shut state, shut resting state. You may remember that in the Del Castillo-Katz mechanism, we had only 1 molecule of agonist, but more importantly, you immediately went to open. That was not the case for the glycine receptor. We could see that there had to be another layer of states here, which we called the flipped state. These states are still closed. The channel is still closed, but how do we know these states are there? We know these states are there because the affinity for the agonist has changed. The affinity for the agonist has increased. So you can have another layer, a layer of, let's say, pre-opening intermediates, in which possibly, I will show you evidence later, that probably the extracellular domain has already changed, has already shrunk around the agonist in the binding pockets, and it is from these states that the channel can open. And you can have 3 open states that are liganded here. Now, I'm going to add these arrows here. So we have resting states, flip states, and the open states. These states are closed. Now, I have added these arrows in gray, not because they don't exist, but because we cannot get direct measurements of these steps. In particular, note that we can have, of course, there are unliganded openings, but they are extremely rare. These channels, are synaptic channels, and they want to keep the signal-to-noise ratio as good as possible. Unliganded openings, the ligand-gated ion channel equivalent, if you wish, of constitutive activity, are very rare in the wild type. Of course, I can release the gate and mutate it and make it possible to measure constitutive activity. And this is how, for instance, our colleagues in the States in Buffalo have estimated that unliganded openings in the muscle nicotinic receptor, one occurs every 15 minutes on average. So that's why we cannot measure them. But you can see, now that I've put in all these arrows connecting the states, you can see a family resemblance. You can see the look of an allosteric mechanism here. If this were just this lot here and had 4 binding states here, it would be a mono-Wyman and Changeux allosteric model for hemoglobin and oxygen binding, right? So, as I was telling you, what we do, we fit this model directly to the data. And because we fit the model directly to the data, we discard all the models that don't work, and this is the minimum model that accounts for all our data. Note also that we are not assuming anything. We don't need to make assumptions about the connection between occupancy and signal, or between stimulus and response. We don't need operational assumptions here, because we're just fitting this model to the data. So we're fitting to the sequence, and I'm going to show you a very busy slide next that shows you how the data displayed as dwell time histograms are very well predicted by this model, which is fitted to the recordings themselves. And the recordings are not shown. What we show here are the dwell time histograms in different concentrations of glycine. These are the open times, these are the shut times. We have other displays, but I didn't want to bore you too much with histograms. What matters here is that there is a very good correspondence, a very good match, a very good prediction of the ragged histogram here, which is the data, by the predictions of the model that are the continuous curves. And this is a result of all our efforts. This is a flip mechanism. So, So what? We have here a bunch of states, we have a bunch of arrows, and a bundle of numbers. What do these numbers, what does this mean? This is a very abstract way of putting, it's a very powerful way of putting our data, but it's a very abstract way. So the next section of my talk, what I propose to do in the next section is to take this mechanism in my hands and twist it around and try to get a mental picture as vivid as possible of what it means to be an agonist in this channel. And I'll try to build up from the abstract mechanism up towards a clear physical structural picture. So, efficacy. In the flip mechanism, take 1. Remember the Del Castillo-Katz mechanism? This time I've drawn it to look like a little bit like flip. And you remember R binds A becomes AR, and you'll have just one step here that goes to the open state. And it is this step that is the step that determines efficacy. Only one step. So the first difference for flip is this. Ouch, this is very big. Let's get rid of all that is not relevant. Let's just look at the right-hand side part of the model where the channel is saturated by the agonist. It's obvious, there's 2 steps now. This is a maximum response here, and we have both equilibrium constants for the 2 steps that determine the maximum response. So we have now the possibility that efficacy resides in flip, in the opening, or in both. What is the case? Well, we didn't know what we would have found. So these are the numbers for glycine. The numbers for glycine show that glycine, which is a full agonist, remember this is, I can't even insert the shut state here because it's open all the time when it's not desensitized. So glycine, when it's bound, when 3 molecules of glycine are bound to the receptor, you will remember, is open all the time. And it's open all the time because it flips very well. 27 means that this equilibrium is 27 out of 28 in this shifted towards here. So it's mostly here. And 20 as an opening equilibrium constant means that of these 2 states, the channel is 95% of the time is in here. What about taurine? Is the difference in both steps? Well, I wouldn't be making such a fuss if it were, would I? The difference is mostly in flip. Now, let's look at it. The opening here is about 10. The opening equilibrium constant is about 10 for taurine. That means that once it has flipped, the channel is 90% of the time open. So there's not such a big difference between 90% and 95%. These, if you also add, the errors in the estimation, these are not really different. Only twofold difference. But these are 200-fold difference. Huge difference in the flipping step, in the initial step. Now, so taurine is a partial agonist because it cannot flip the channel. Now, let's do another take. Can we get a little bit more concrete and physical? Can we grapple with the reality of this channel? What does it mean, for instance, in terms of the time spent in the different states, right? Okay, so resting state, A3R, and we've drawn a cartoon here in which we have the binding site all loose and open around the agonist. Flip, we assume that the extracellular domain has changed, channel is still closed and open. Glycine is a very good agonist because on average, it spends a tiny amount of time in the resting state. You see, 50 microseconds is about 0.2% of the time spent in the saturated state. Then it flips. Flips is a very short transient. It's about 8 microseconds. And it took all the power of the improved analysis that we had to be able to do this and measure it. But most of the time, glycine, the channel opened by glycine is open. The channel activated by glycine is open. What about taurine? Taurine is a poor agonist because it lingers. It dawdles around. It's a lazy chap. The channel bound to taurine lingers in the resting state. This is 1.5 millisecond. So this is a very long time compared to taurine. So 1.5 milliseconds versus 50 microseconds. But once it's flipped, you see, the flip state is not that different in lifetime because the flip state is mostly dictated by the speed with which the channel can open there. Right. So, can we make efficacy take 3? I haven't shown you yet anything about the physical structure of these channels. But of course, in the last 20 years, there's been an enormous progress in structural work. We now know this is torpedo, receptor, the work of Nigel Unwin in Cambridge over decades of averaging cryo-electron microscope pictures has shown this picture here, which is the latest refined picture. And the channel is a pentamer, and I'll show you a single subunit, just much clearer. So you can see a transmembrane domain with 4 transmembrane segments, and an extracellular domain here, and a cytoplasmic loop. The binding site, we now know for sure. It's at the interface between subunits in the extracellular domain. Another stroke of luck, there is mollusks have in some of their synapses a soluble form of this extracellular domain. These proteins, the acetylcholine binding proteins, were crystallized in the early 2000s by the group of Breach and Sixma in Holland, and this is what they look like. This is 2 of them. This is a principal subunit here, like that, and the complementary subunit. The pore is towards the back of the slide. If I take away the complementary side subunit, you see carbachol bound behind this loop here. This is the C loop. And this is kind of an interesting loop. It's one of the loops that were predicted by work with photoaffinity labeling in the 80s and 90s by the group of Changeux and the group of Cohen in the States. It was predicted to be one of the binding determinants. So this is particularly interesting among all the loops that are in the binding site because other work on the acetylcholine binding protein shows that with activation, this loop may move. Now, why? Well, you can look at the structure of this when the acetylcholine binding protein has nothing in the binding site, or an antagonist in the binding site. So in that case, this loop either flaps in the breeze here and is open there, or if it is an antagonist, it's stuck. It's stuck in the open position. But if an agonist is bound, this loop closes in, so the structure of the binding site looks quite different. So what can we sort of imagine is happening in terms of structure when we look at efficacy by an agonist? How do we see efficacy? So let's try to think a little bit from the structure back. Now, this is the structure. Now, okay, so ligand-gated ion channels are more like switches, perhaps, than integrators and complex machinery, but they're not that simple a switch. What about a ship's throttle? You have a full agonist is a full agonist, perhaps, because he can move the C loop an awful lot. You have a huge movement there, and this huge movement there goes down all the way, and it's a huge movement in the transduction area here, and it is because you have a big perturbation throughout the chain that the agonist is a full agonist. Conversely, maybe taurine is a partial agonist because it only goes, barely ahead, not full steam ahead, but barely ahead, just a small change here, a small change there, and therefore partial agonist. So, if this is the case, we would imagine that the intermediates that we find along the way are different for full agonists and for partial agonists, right? Can we get information from our fit of the flip mechanism? So, efficacy and the flip mechanism take 3. This time we got to look at the agonist efficacy as the agonist hand in hand with the channel go over the energy landscape. So the channel and the agonist have become mountaineers, and they're going on a path of energy. You remember that we've seen a slide in the Gadam lecture with an energy diagram, an energy landscape in 3 dimensions. But what we're going to do is look at a single path there for ligand-gated ion channel and see what our direct measurements of this path say. Now, so this time I'm sorry, I put it on horizontally to show you, because I tend to think of mountains as going up. So here we have resting, flipped, and open. What does the pathway look like? This is glycine, and glycine starts at resting here, goes over a path. This is the activation energy that determines the on-flipping rate constant, goes over this path, but then the flipped state is downhill from the starting step. The open state is also downward. So there is an activation energy, but glycine is down all the way. What about taurine? That's an interesting thing. Will taurine differ in both steps? No. Taurine differs almost exclusively in the first step. Once it gets to this flip state, the opening is kind of similar. So that doesn't agree with my ship's throttle commanding a big perturbation throughout the chain of transduction idea. Now, of course, this is just 2 agonists on the glycine receptor. Is this a quirk of the glycine receptor? No, it isn't. Now, I want to show you the next slide, because it's a very crowded data slide, but it's years and years of work. This is back to the muscle nicotinic. This is a work of Remy Zlape in my lab, really a cornerstone of my lab. And these are single-channel dose-response curves obtained for the muscle nicotinic, cleaned of agonist block when you are at negative potentials, or obtained at positive potentials. We have 4 of them: acetylcholine, TMA. At positive potentials, acetylcholine has become a partial agonist, and TMA is even more partial. If we look at the energy diagram for these guys, what do we see? Well, I'm going to throw in also choline, another paper. So these are 3 papers summarized, and they look exactly the same as glycine, don't they? The difference between the agonist is a difference in the flipping step. Once you flipped, the opening is kind of similar. So this would argue, and it is a little bit baffling, this would argue that it is, once you got to flip, the flip states are roughly similar in structure because they open pretty similarly. So the agonists are different because they're different in stabilizing this flip state. Now, this is a work to 2008. How do we move on from here? Now, one thing that we have done, and I don't have time to show you, we have looked at all possible variants of the glycine receptor. And we have found that we need flip, we need these intermediates to explain the activation of these receptors throughout the glycine isoforms. Other people, most notably the lab of Steve Sine at Mayo Clinic in Minnesota, and the lab of Tony Auerbach more recently, have also needed to insert pre-open partially activated intermediates in their mechanism in order to account for their data in channels in this superfamily. Steve Sine in 2009 called it primed. Tony Auerbach now 2 years ago called it catch and hold. The concept is the same. They need the intermediate. So how can we pursue the investigation of what is the structure of flip? Remember, the flip is also a very, very short intermediate, 8 microseconds. Can we kind of slow it down a little bit, perhaps, and maybe it will be easier to explain? Work from now on is all going to be work in progress that shows you the different strategies that we are pursuing, slowly and painfully, towards progress. Now, let's go back. You remember our Startle disease mutation? What, almost 20 years ago now. K276E. Now, we can look at that again. And this time, we will look at it by another technique that has become available, and that is a technique that applies to outside-out patches, very brief, saturating pulses of high-concentration agonist. So this is done by a theta tube. This is the control stream or very high concentration. This is either 3 millimolar or 30 millimolar glycine. And you can see that this pulse produces a current. This current, incidentally, I don't have time to go into that now, but this current is an excellent proxy. It's an in vitro pseudosynaptic current, because this pulse here is exactly what happens at the synapse, at the neuromuscular junction, but also at most central synapses. So this is a way of measuring a synaptic current in vitro. So what happens in the wild-type glycine receptor is, you see here, we got a very fast onset of the current, and we have about 7 millisecond decay. Now, the onset of the current is so fast that, in fact, it's limited by the speed of our pulse. So we can't even measure how fast the channel is, really. But if we mutate the channel and we take the K276E mutation, what we find is, I've scaled these, we find that the rise time now is much slower. Here we have it 1.3 millisecond, and because of what I was telling you, this is not a real measurement, it's actually an overestimate of the speed, of the rise time. The real rise time is much faster than that. So there is a major slowing of the channel on its way to activation, and this is because we have damaged flipping. We have damaged flipping, and we have made also the open state less stable. You can see this channel is now open for a shorter time. So we are confirming here that this mutation, as we hypothesized in 1998, is indeed a gating mutation. We were right. So we can obtain a dose-response curve for this mutation, a single-channel dose-response curve, and now glycine has become a partial agonist. No surprise there. Now, I'm going to show very briefly, just flash for you, the mechanism that needs to be fitted, and this time, because we are slowing, this is a channel in slow motion that is reaching the open state, stumbling along the way, clunk, 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 struggling, we need several layers of flip. What matters is that flip has now become 3 states, one of which is twice as slow, and it is possible that we may be able to use this mutation to slow the activation of the channel and be able to get a better grasp on flip. Of course, ideally, what we would like to do is to have the structure of flip. But members of this superfamily are not that great at giving structures. There are good structures that are coming along, but one structure, for instance, is that of the prokaryotic homologs of this superfamily, such as Glyc and Elic. Indeed, we'd like very much to do work on these channels, particularly Glyc, because 2 years ago, this is a paper by Corringer and Delarue in France, and by trapping here, this is the extracellular domain, this is the channel, these are the transmembrane domains, and this is a transduction interface. What they've done is put in a cysteine mutations, and they managed to covalently freeze the channel in a conformation that is neither resting nor open. They called it locally closed. So it's a conformation that is a little bit activated outside, and it's still closed inside. Sounds like flip, doesn't it? We'd dearly love to look at this, and we tried. But, this is another one that hits the dust, unfortunately. Not all channels are amenable to our sort of work. Glyc, unfortunately, has got tiny single-channel conductance. So we've mutated everything that we could conceivably mutate, and we managed to increase the conductance from 8 picosiemens, that is terrible, to 12, which is about pathetic. So we can't work with this. In addition to that, the binding site of this channel, well, this channel is activated by protons, damn it. So protons are likely to bind in a sort of more delocalized way than agonists do, so it's not a great channel for us. Pity. So we tried, in collaboration with Raimund Dutzler, who's a crystallographer in Zurich, and interested us in another channel, another bacterial channel. This is a channel from Erwinia chrysanthemi. I hadn't heard of it before it came up, no. But it causes, despite the chrysanthemum thing, it causes soft rot in potatoes. There you go. The channel there is a big channel. It's got a nice big conductance. And when we started working on that, we thought, right, this is a channel for us. This is a channel on which you can do structure and you can do fine function. So the other good thing about this channel, well, it's only seen here because you can see how slow how big this calibration bar is. But the limiting on rate here is 10, 20 milliseconds. Remember, for glycine, it was unmeasurable, effectively, because it was less than 0.2 of a millisecond. So they're slow. That's good. That's good, isn't it? We've also looked at this together with Raimund, and we were looking at modulators in the transduction area, with ionic modulators, divalent cations, and we found that this slow activation is due to the fact that there is a flipped state in the activation, and that access to the flipped state is slow and limits the speed of activation. However, this channel, you can see the red arrow there, and those of you that are electrophysiologists will recognize an off-current. This is a cationic channel that gets blocked by the agonist. So that's bother. And another thing here is this channel has undoubtedly got big open conductance, but you can see here that, well, the high concentrations look very much like what we were seeing before, but you can see that you have fairly short clusters here, and you have fairly long clusters at low concentration. So the concentration dependent is not as striking as we had before. For those of you who are single-channel aficionados, what's happening here is that the deactivation and the desensitization time constants are such that each clusters contain 1 or 2 bursts. This is not ideal. We weren't able to get a p-open dose response curve, in fact. We've analyzed this channel, and the paper is now coming out in Journal of General Physiology soon, but it's not an ideal channel. I think that what we'll have to do, and we're working on that, we are going to engineer this channel so that we can get its rate constant, its energy landscape, back in the sweet spot for single-channel kinetics, and that's our best hope for ELIC. But why not go back to the glycine channel? The glycine channel, on the face of it, has got a lot going for it. It's amenable to single-channel recording very well. It's got lots of agonists. It doesn't get blocked by the agonists. Of course, we don't have a structure for the glycine channel, but there are structures for GluCl, which is a C. elegans channel that is reasonably homologous to glycine channels. So, efficacy, last take, back to the glycine channel. This is a homology model, and this is work that we've done in collaboration with the lab of Phil Bigging in Oxford. And this is work by Riley Yu in his lab. You can see here you have the homology model based on GluCl, extracellular domain, glycine in the binding pocket, transmembrane domain. So you can see, I can show you as many people, I mean, there's a flourishing literature on homology models, and we can show a lot about what is called agonist recognition, which is a good sort of compromise word, perhaps. So the agonist recognition shows glycine in the binding site. You can see here the C loop on the principal side. This is glycine there. You can also see a molecule of water. Interestingly, here you can see a short list of residues that are important for the binding of glycine. In particular, there is the famous aromatic box that has been described for practically all receptors in this superfamily. If we tilt this slightly, you will see more easily the water. That is a unique feature of this, of our homology model, and the glycine here. Now, can we connect the numbers, the data, the thermodynamic constants that we have got from our analysis of function to this structure? So we'll have a last take at looking at flip. So we were emphasizing before the flipping and the opening. This time, we are looking at the binding site. Now, of course, the binding and gating problem remains, but there has to be efficacy in the binding site, because that's where the agonists bind, and different agonists have different efficacy. So what does it mean to be a differently efficacious agonist up there in the binding site? Well, instead of measuring the flipping conformational change and the opening, we are looking, well, let's focus on what happens to the affinity for the resting state and the affinity for the partially activated flipped state. You can see here that the affinity goes up quite a lot for glycine, and indeed it's 65-fold here. For taurine, the affinity barely changes with activation. So the highly efficacious agonist glycine increases its affinity by 65-fold, and this is the equivalent of 2.4 kilocalories per mole, almost a hydrogen bond. Now, what does this mean structurally? Can we connect this to that? Well, it's difficult, because we don't know what this is. Is this resting? Is this partially activated? Is this open? We don't know for sure. And it's difficult just to relate just to the binding affinity and guess like this. There is a way forward. It's a slow and painful way. There's a surprise. Let's just look at it in the last slide. We can have a map of our bonds, of the bonds between the agonist and the binding site here. What we can do is try to measure, with our techniques, the interaction, the direct interaction between parts of the molecule of the agonist. Say this is a carboxyl, and it interacts with that arginine there. How do we do that? There is a technique, it's called double mutant cycles. It's a well-established thermodynamics technique to measure the coupling energy between two bits of the molecule. What it requires us to do our measurements in control conditions, one, to do it when we mutate the amino acid, 2, to do it when we mutate the agonist, 3, and to do it when we mutate both. So it's tough. But it is in principle doable. And that should allow us to tell you which of these bonds change with activation. So, finally, how do we mutate an agonist? Quite simple. We call it structure-function activity, don't we? This is the carboxy terminal of beta-alanine here that becomes sulfate in taurine and sulfite with hypotaurine. We've already done beta-alanine, we've done taurine, we're working on the mutants now. So, conclusions. I hope I've been able to show you that we need pre-opening intermediates to understand the activation of these receptors. Pre-opening intermediates have been discovered and been detected in all nicotinic superfamily channels analyzed so far. And what is very comforting is that it's been done with different techniques, different software analysis. Whether we call it flip or primed or catch and hold doesn't really matter so much. Agonist efficacy is determined in these channels by access to the early conformational changes, that is, access to flip. We've shown that in the glycine receptor and the nicotinic receptor ourselves. Last week, there's been a lovely paper, I'm very pleased about it. It's a paper by Cecilia Buzat in Argentina that has shown on the 5-HT3A receptor, again, completely different software, completely different analysis, that this principle is valid also in 5-HT3A receptors, which are other receptors in this superfamily. I've shown you that we can measure the increase in agonist affinity with activation, and this is the new structure function for our days. As pharmacologists, we live in very exciting times. On one side, we have more and more structures becoming available for our targets. We have molecular dynamics, increased computational power, increased software ability to understand the dynamics, to simulate the dynamic of the receptor. But I hope I've convinced you that our work shows the ligand-gated ion channels are a privileged testing ground for our hypothesis of agonist efficacy because they allow us to measure the molecular counterparts, the physical constants that determine and describe these molecular changes in activation, and hopefully to relate them closely to the structure of the receptor. The most important slide is this one. These are the people that have worked with me since I started my lab. You've seen work by Valeria Buzzomato and Marco Beato. They've now left my lab. Andrew Plested worked on the K276E channel. These are the people that are in my lab. It's invidious to choose any of them, but Remy Lappe has been involved in this project from time zero. My collaborators, I wouldn't have got anywhere without David Cahoon's and Alan Hawke's advances in the 90s, and without his help and support with the software analysis. We're working on refining our methods with Bayesian analysis in collaboration with Mark Girolami. I haven't had time to show you. Raimund Dutzler in Zurich for ELIC, Phil Biggin in Oxford for the molecular dynamics. BBSRC funds me for ELIC, but all the work that you've seen has been funded more or less continuously by the MRC since 1997. Thank you.